We've been waiting for you. That's what I said. Didn't I say that? All right, guys, giddy up. Let's do it here. So, that's my question. All right. So there's, I'm in the dock here, to quote Al Pacino. There we go. All right. So, where's like, is the chemistry something going on, or? It's 72 degrees outside. Is that it? All right. All right. Really? All right. Let's get into it, you guys. You're yeah. All right. So, 4 requirements of DNA as a genetic material, right? 4 structural requirements. Okay. The first was its ability to encode vast amounts of information, and we saw that it accomplishes that because there are no restrictions in the order of nucleotides along the length of the molecule, right? So we saw that as we build the molecule, come on, there we go. There are no restrictions in how we assemble the molecule along its length, and because there are no restrictions, it means you can have an infinite sequence of nucleotides built along the length of the molecule. Right? Across the molecule, it's rigid, isn't it? By way of the specificity of base pairing, all right, where A always pairs with T by way of 2 hydrogen bonds, and G always pairs with C by way of 3 hydrogen bonds. So that gives us that specificity of base pairing, okay? That gives us a predictability. If I give you one strand, you can tell me what the other strand does, all right? It also sets us up for what we're about to talk about, which is that second requirement. And the second requirement is the molecule must be capable of self-replication, of copying itself, all right? And we're going to see then when we say that, we mean that what? Each half of the double helix, each side of the double helix is going to become a template for making a nascent meaning a newly formed strand, all right? So each original strand will form as a template, or at least that's what Watson and Crick were proposing as they described the structure of the molecule as a double helix, because apart from that idea, there's really no reason for DNA to be a double helix, all right? All you need is one strand as your coding strand, and that's, you'd have what you need in terms of its role as a blueprint, right? But it exists as a double-stranded molecule, and the only way to explain that, according to Watson and Crick, was that that's essential for self-replication, where each half of the molecule will become a template for building the other half, all right? And in that way, you always preserve the coding strand, all right, or the coding information, all right? You don't get an inverse of it one time and the coding strand the next, all right? Okay, so, pardon me, where we left off last time then was to consider their proposed model, which was a semi-conservative model for replication, with the other 2 options. And I want to quickly, now especially since we're off to a late start here, is just show you the experiment that was done to verify the Watson and Crick's hypothesis, all right, of self-replication. And so Messelson and Stahl did this great experiment. Let's just put it all up there. They grew bacteria on a medium containing a heavy isotope of nitrogen, all right, N15. And what they're about to do is they're going to take the bacteria that have been grown on that, they're going to break them open, and they're going to run their DNA through what's called a density gradient, all right? So instead of centrifugation, where I taught you earlier about differential centrifugation, you're centrifuging things down, and what comes to the bottom of the tube? The largest things, right? It's centrifugation by size. That's differential centrifugation. And remember, we separated out the different parts of the cell, right? The nucleus, and then the mitochondria and chloroplast, and then the microsomal fraction with the other membranes. Remember? All right. We're going to look at a different kind of centrifugation. It's called isopycnic centrifugation. It is centrifugation or separation on the basis of density. Okay? And you're not going to run things down to the bottom of a tube. Instead, you're going to create a gradient. All right? So the material that we use for centrifugation is called cesium chloride. All right? And we're going to have a cesium chloride gradient, so that instead of a solution of cesium chloride, it's actually getting more concentrated as you move closer to the bottom of the tube. All right? I think you get the idea. All right? When we put something in the centrifuge, and here we're going to be using the ultracentrifuge, we're going to be spinning not at 700 RPMs or at 1200 RPMs or at 5000 RPMs like we would if we're doing differential centrifugation. We're going to be spinning at 60,000 RPMs, all right? And the molecules that are layered on the top of the gradient will begin to move down through the gradient, all right, through the centrifugal force, and they will band out at what's called the isopycnic point, all right? The isopycnic point is the point of equal density. So they're going to move until they get down to a region of the gradient that's the same density as the molecules themselves. You can keep spinning it. If you were doing differential centrifugation, eventually everything goes to the bottom of the tube, doesn't it? But here, it'll spin until it finds its isopycnic point, the point of equal density, all right? And it'll stop there, okay? And so if I take DNA that's been grown in, or bacteria rather, that have been grown in heavy nitrogen, remembering that part of the DNA molecule is a nitrogen-containing base, all right? Then the DNA contains this heavy isotope of nitrogen, and it's going to band out at some point in the tube, all right? We'll call it here, and we'll call it a heavy band, all right? It is the DNA that is carrying heavy nitrogen, all right? And so what these guys did in this experiment is they grew DNA for one generation on or they grew it for multiple generations, pardon me, until all the DNA had the heavy isotope of nitrogen, all right? And then they grew it for one generation on a light isotope of nitrogen, all right? So let's consider the three different models for DNA replication. I'm going to go back here, all right? Let's consider the three models. Conservative, semi-conservative, and dispersive. Okay? Starting with the same. We've grown them for a long time on heavy nitrogen. We run it down. All tubes are going to look like this, a heavy band. Right? Okay. Now I grow it for one generation on light nitrogen. If DNA replication is conservative, then I'm going to have my original molecule, and I'm going to have the new molecule. I've grown for one generation, right? And so one of the strands is going to be the heavy. So what's it going to look like if this is the model I'm going to get in that new run? I've grown them for one generation on light. I'm going to get a heavy band, and I'm going to get the light band, reflecting the DNA molecule that was copied now using light nitrogen. Okay? If semi-conservative replication applies, then what am I going to get? Semi-conservative, each strand is made up of half old, half new, meaning half heavy, half light. So I'm just going to get a medium band. Right? And if, likewise, I replicate the DNA, but I use the dispersive model where I'm making twice as much DNA as I had before, I'm breaking down all the nucleotides and reconfiguring them with some light, all right, I will also get a medium band, won't I? All right, so the result of that first round here, having grown on light nitrogen, of the semi and the dispersive is going to give me the exact same result in terms of where they band out. All right? When I do this experiment, I don't get this. All right? I get a medium band. So then I grow them for another generation on light nitrogen. All right? And if I grow for another generation, and you look at our figure here, dark blue is the original heavy nitrogen, light blue is the light nitrogen. All right? It makes sense what we're going to get. We're going to get another molecule that's a hybrid. We're going to get this one that's going to be completely light, right? Here again, another one that's a hybrid, medium, and another one that's completely light. So in that next generation, if it's semi-conservative replication, I'm going to get a light band and I'm going to get a medium band. Good? Everybody see it? If it's dispersive, what am I going to get? They're going to be all sort of a mix, aren't they? And the more I do this in light nitrogen, the larger proportion of each DNA molecule is going to be light, right? And so in this next generation, I'm going to get what? A single band that is medium light, right? I'm increasing the proportion of light nitrogen in all replicates, right? That's what I got, all right? This is the result they got. So it demonstrates then DNA replication is semi-conservative. And those are the results you see there, all right, as this is what we just did up on the board here, okay? You didn't get this result, you didn't get this result. This is the result you got, all right? It confirms that DNA replication occurs semi-conservatively. Right? So that's what we want to look at today. We want to look at the mechanics of DNA replication. And what do we know about DNA replication already in terms of as cell biologists, not molecular biologists yet. As cell biologists in bacteria, you know that what? That in bacteria are always replicative, right? They're always reproductive, right? As soon as they finish replicating their DNA, they divide, binary fission. And once they divide, what do they do immediately? Immediately begin replicating their DNA again in advance of dividing. There's no vegetative state. There's no stop. There's no point where bacteria stop replicating their DNA. In fact, under optimal conditions, they're replicating their DNA. And before they even finish dividing, they've begun replicating it in advance for the next cycle, all right? And the generation time is dependent simply on how long it takes them to replicate their DNA, which is a function of the environment providing them with what they need, all right? So generation time is just, do they have everything they need to replicate their DNA? And if they do, they can do it quickly, and they just keep dividing, all right? In eukaryotic cells, what do you know? When does DNA replication occur? Guest on Friday. Yeah. During the S phase of the cell cycle, right? During the S phase, remember, where's the S phase? After G1, just before G2, in the middle of interphase, right? In the middle of interphase, long before we get to mitosis, right? Okay, and so you've got replication happening in both prokaryotic and eukaryotic cells, all right, a little bit differently, because once you replicate in eukaryotic cells, you're going into G2, right, and then divide, and it may sit, once it divides, it may sit for a long time, right? You have vegetative life there. Okay, all right, so let's look at DNA replication. And we'll start by simply taking our bacterial genome here, as you see it, right, a circular loop of DNA, and we'll note that in prokaryotes, DNA replication occurs from a single starting point. There is one origin of replication, and it's going to occur in both directions from that origin of replication. So the circle I've drawn here on the board represents the double helix. Even though I've just drawn it as a single line. And so if I now show you this, all right, I am copying from the origin in two directions. I'm replicating bidirectionally, all right. Eventually, I'm going to end up with that, right? And eventually the two will separate completely, right? We'll end up with our two pieces of DNA, right? We're going to zoom in on this right here, okay? And I show you that because that's what's shown on the screen here, but I want you to get your bearings here, all right? What you're seeing here is replication going in bidirectionally from our point of origin, and each of these then green boxes represents a replication fork. All right, and I want you to see that replication fork on the screen then is what you see here. So that which way is replication going? Upwards on the screen here or downwards? Upwards. We're moving towards the fork. That's why I drew that, all right? Does it make sense now when you stop and think about it? It's doing this. It's going in that direction, right? That's what we're doing here. This is our parent strand. And replication is moving in the direction of that, and we are now generating our copies. You have to climb into what we just finished talking about, which is hard to do and worth taking a moment to do. So this is the original parent strand, yes? And this is the new nascent strand, all right? This is your new strand. There's your semi-conservative replication. Likewise here. Here's the original, all right? And I'm moving in that direction towards the fork, in the direction of the fork, all right? Here's my old original, and here's the new part that's being synthesized, okay? Everybody's good? So you have to kind of look at it and make sure you get it in your head, all right? And so here you see, again, our DNA replication taking place in our prokaryotic cell. A single origin of replication, and replication under ideal conditions takes 20 minutes, all right? So there it is. There's our 20 minutes, okay? All right. Well, you've got 1,500 times the amount of DNA that's found in a prokaryotic cell. So how long is DNA replication going to take for you? Well, don't do the math, because there's a difference between prokaryotes and eukaryotes, many, but we'll identify one. Here in prokaryotes, you have a single origin of replication, with replication happening bidirectionally from that point of replication. And when those two replication forks meet here at the bottom of the molecule, the two molecules will separate, and we'll have two circular loops of DNA, right? They probably will have each already started to replicate when we separate them, all right? Talk about getting ahead of yourself, which it is doing. And when these meet at the bottom, obviously, as I say, the two loops of DNA can separate. In eukaryotic cells, you can have multiple origins of replication. Each is occurring bidirectionally, all right? So instead of a single origin, each chromosome, all right, we think of chromosomes in eukaryotes as what? They are, what have we said about the eukaryotic genome? It's larger, it's diploid, which we won't worry about right now. It's a fragmented genome, meaning that the whole genetic blueprint is divided into chapters, chromosome 1, chromosome 2, chromosome 3, all the way up in humans through chromosome 23, all right, diploid, 2 copies of each, all right? And for each of those chromosomes then, there it is, double-stranded DNA, there are multiple origins of replication, which you see here on the figure, all right? Multiple origins. Replication will occur bidirectionally from each of those. And each of those then replicated forks. Here's your replication fork. There's your arrow. Make sense now? Right? There's your replication fork. There's your arrow going into the fork. Okay. and that's going to collide with replication coming from the adjacent origin of replication. So even though you have 1,500 times the amount of DNA, you can replicate your DNA in an hour and a half. That's pretty cool, all right? Right, because you have hundreds or even thousands of origins on a given chromosome, all right? So I think you get it. I don't think I need to belabor that any further. Any questions on that? Okay, so there's replication then. As we look at it just sort of from a bird's eye view, let's zoom in now and start talking about what it's entailed in order to replicate at the replication fork. So we're going to focus in on a replication fork. If we had opened up an origin of replication here in our eukaryote, right? Again, DNA replication is moving away from the origin, all right, in the direction of the replication fork, and so we are going to be looking at this, right? We're going to be looking at a replication fork right now and what happens there, okay? And let's think about a few things here, all right? We have said that each strand of the double helix is going to serve as a template. All right, so right now we're going to be just looking at one of the two strands, one of the two parent strands, which is what you see on the screen right now. This is one of the two parent strands, and we're going to be adding new nucleotides to that. All right, the nucleotides themselves exist in the cell as what we call nucleoside triphosphates. All right, and so we could call, for example, ATP a nucleotide, or we would call it a nucleoside triphosphate, which simply says the 5-carbon sugar and the phosphate group, or sorry, the 5-carbon sugar and the nitrogen base have 3 phosphates attached to them. So we call it a nucleoside when we are not including the phosphate groups, or we call it a nucleotide when we include the phosphate groups in the molecule. So I'm going to call this a nucleotide or a nucleoside triphosphate, recognizing the 3 phosphates, and of course the other 3, ATP, GTP, TTP, and CTP. The raw materials for replicating DNA exists in the cytoplasm of the cell, all right, as triphosphates, okay, sorry, in the nucleoplasm of the cell, as nucleoside triphosphates, all right, and these are going to give up a pyrophosphate, meaning we're going to break off both terminal phosphates and end up with AMP, and likewise GMP and TMP and CMP. And when we cleave the 2 phosphates at the same time, we call that pyrophosphate. That is an exergonic reaction which can be used to pay for the addition of a covalent bond between neighboring phosphates, all right? Or between neighboring nucleotides, sorry. All right, and so that's what you see here on this figure. We're using our template strand. We're allowing nucleotides to pair spontaneously. So A is going to pair with T spontaneously by way of the specificity of base pairing. I don't need to be coordinating that. It's just right there built into the chemistry. All right, these will pair spontaneously, and when I cleave the pyrophosphate, that will pay for the formation of the covalent bond between the sugar of one and the phosphate of the next, right? It's a covalent bond. We said it was a phosphodiester covalent bond, sugar to phosphate, and we're paying for then the formation of that bond. Here it is, between the sugar of this and the phosphate of the next, covalent bonds along the length of the molecule and across the molecule, of course. They've paired by way of hydrogen bonding, okay? And we're paying for that covalent bond by way of pyrophosphate cleavage. So the raw material for replicating DNA is nucleoside triphosphates, ATGC, all right? As we cleave pyrophosphate, they become our nucleotides, meaning what? Is when you drew your nucleotides before, you did what? A 5-carbon sugar, a nitrogen base, 5 carbon here and our phosphate group. And there's your nucleotide, isn't it? Okay. In the case of DNA, it just has a hydrogen here at the number 2 carbon. In the case of RNA, it has an oxygen there, as we've said. All right. And we'll recognize very quickly here, there's our number 5 carbon, and here's our number 3 carbon. All right, with its hydroxyl group on it, okay? There's our number 3 carbon, all right? And that allows us to begin to recognize, as we've said before, the polarity of the molecule. So as we talked about the molecule yesterday, we mentioned that here's the double helix. The 2 strands run anti-parallel, all right? And we can recognize that polarity, all right, meaning the directionality of the molecule, by recognizing again here our 5' carbon here. We call this the 5' end of the molecule. This is the 3' carbon here extending down at the end of our strand of DNA. So there's our 3' end. And you see that the complementary strand is inverted, right? And so one strand runs 5' to 3', the other strand runs 3' to 5'. Jessica? Good? All good? Okay? All right. So, if we get to the replication fork now, all right, we encounter an interesting problem. Here's my replication fork. Okay, which way is replication happening? It's going in this direction. Let's put a big arrow here so we know that's the direction we're moving, okay? All right, as we replicate. And let's label the molecule in terms of its polarity. So I'm just going to call this the 5' end, and this is the 3' end, and therefore this is the 5' end, and that's the 3' end, okay? Good? All right. Replication. We're going to replicate both strands, aren't we? And the overall direction of replication is toward the fork. However, the machinery responsible for DNA replication can only read the molecule in the 3' to 5' direction, which works fine for this strand, all right, for the strand on the left here, all right? As we replicate, we're going to read 3' to 5'. This is the way that we read the template, all right? So this is the reading direction on the template, okay? We read 3' to 5', which means that as I'm laying down my new piece of DNA, right, semi-conservative, we're going to copy each of the 2 strands, aren't we? I can replicate this strand and lay down new DNA 5' to 3. Synthesis is going to be 5' to 3'. If I've just said we read 3 to 5, then replication is going to be 5' to 3', right? If we go back here for a second, all right, recognize that the new nucleotide is being added to the 3' end, isn't it? Everybody's good there? The new nucleotide is being added to the 3' end of the one that's already bound. Okay, now you may be anticipating where I'm going here. What do I do about the other strand? We read 3 to 5'. This is a 5' end here. Which way is the fork going? We're replicating up the board here, right? How do we do this? We're going to have to replicate in small pieces, right? We're going to read 3 to 5' and make a little copy. And once we do that, we're going to do it again over here. And then we're going to do it again over here. All right? We're going to replicate in small fragments. And these fragments are called Okazaki fragments. Okay. And the Okazaki fragments in prokaryotes are actually much larger. In prokaryotes, they're 1,000 to 2,000 nucleotides in length. And in eukaryotes, surprisingly, they're only 100 to 200 nucleotides in length. Okay? So there's a pretty big size difference in terms of the size of the fragments themselves. But we're going to lay down a fragment, and then we're going to lay down another and another. So what's my net direction? My net direction is still up the board, isn't it? I'm putting this fragment in, and then this one, and then this one. And so my net direction is still in the upwards direction here. And we're going to call this new strand that's being produced as one solid piece, we're going to call that the leading strand. All right? And the strand that's being copied or laid down using Okazaki fragments is going to be called the lagging strand. Okay? So you've got the overall direction of the replication fork. We've said we're reading the template strand 3 to 5 prime. We've identified the leading strand on one side of the double helix and the lagging strand on the other side of the double helix. All right. Elongation occurs in the 5 to 3 prime direction. Here it is, 5 to 3. All right. And we either have continuous, which is your leading strand, or you have building it in pieces here using Okazaki fragments to build the lagging strand. Okay? Now this takes a lot of machinery, right? And it's happening really fast, all right? You're laying down about 500 nucleotides per second, all right? 500 nucleotides per second. So the double helix is being opened up, all right, and replicated at a rate of about 500 nucleotides per second. Now, if you remember, there's 10 nucleotides per turn, right? We said for a turn of the double helix, there's 10 nucleotides per turn. So that means 500 nucleotides per second means 50 turns per second, right? Or 3,000 revolutions per minute, right? You think about your car, right? You're going up a hill, you're in third gear, it's about 3,000 RPM, all right? That's spinning, all right? This is helicase unwrapping the DNA at 3,000 revolutions per minute, all right? Uncoiling it and coiling it back up again, all right? So the DNA has to be opened up, replicated, and then it closes. The strands separate, but you're uncoiling that at 3,000 revolutions per minute. Yeah. Well, collectively they're about the same, so they're going to be the same. So they have to keep up with one another, but they're happening in a very different way, aren't they? All right, so now let's take a look at the machinery here. All right, here you see the enzyme that's going to be responsible, or a couple of the enzymes. We're going to get into those in just a sec. All right. The enzyme that's responsible for replication is called DNA polymerase, not surprisingly. All right, DNA polymerase, the enzyme that's polymerizing DNA. But it requires a number of other enzymes, all right? And so let's take a look at what's required here, all right? First off, to open up the double helix, we need what's called helicase. All right, helicase is an enzyme in both prokaryotic and eukaryotic cells that opens up. It destabilizes the hydrogen bonds between the two halves of the double helix. All right, so we have one enzyme, helicase. All right, once we open it up, we've got to stabilize it so it doesn't close up again. So there are a bunch of proteins called single-stranded binding proteins. All right, single-stranded binding proteins that bind to the DNA and prevent it from reannealing, from coming back together again. Okay. Now we're ready. We've got our strands separated. We've got our replication fork in place. we can begin to work on the replication process. Replication is going to depend on the enzyme DNA polymerase. But DNA polymerase cannot bind to the DNA and begin to replicate it unless there's a small primer there. The primer is made of RNA, right? And so another enzyme comes into play. That enzyme is called primase. So we've opened up the double helix with the help of helicase and single-stranded binding proteins. Single-stranded binding proteins. Primase binds. In the case of the leading strand, can we say that primase binds at the origin of replication? I should have given this a name. By the way, this whole bit here is called the replication bubble. All right? And of course, there's our origin. And so on our leading strand, we're going to lay down a primer. And then DNA polymerase is going to take off from that primer. Okay? On the lagging strand, I'm going to have to lay down, let's put some primers on the board here. All right. Primase will lay down a primer made of RNA. And then DNA polymerase, specifically DNA polymerase 3, all right, is going to work off of that primer and extend, forming our leading strand. What has to happen on the opposite strand? We're going to have a primer, and then DNA polymerase will work off of that, and then another primer, and then another primer, right? So each time, primase has to come in and lay down a primer, and then elongation occurs, reading in the 3 to 5 prime direction, as we've said. Laying down synthesis occurs always in the 5 to 3 prime, meaning the new piece is going to be 5 to 3 prime. Here you see it on the figure. Here's our primer. Here's the newly made DNA with the help of DNA polymerase 3, all right, which is extending off of that primer, okay, all right. Then DNA polymerase 1 comes in and chews up the primer, and then an enzyme links these pieces together called a ligase, right? Ligase ties together the pieces of DNA, all right? So again, we begin with primase, either at the origin, if we're about to build the leading strand, or at each point in increments here, if we're going to build Okazaki fragments. DNA polymerase 3 can begin synthesis using that primer as a starter, all right, and can carry out synthesis of each of the two sides, all right, again in fragments on the lagging side, in one continuous piece on the leading side. And then DNA polymerase 1 comes in and removes the primer and replaces it with DNA, and then the pieces are then sewn together, connected together with the help of what's called a ligase enzyme, okay? So here you see all those pieces in play, and we can sort of talk about what we've just said, looking at it in a progression here, okay? Here's our leading strand, right? We're just simply reading 3 to 5 here, the top of the figure, leading strand. We're laying down new DNA, adding new nucleotides to the 3' end, right? As we add new nucleotides. We've got helicase unwrapping. We've got our single-stranded binding protein stabilizing. DNA polymerase 3 started up here at the origin and has been laying down new material, reading 3 to 5, adding 5 to 3, all right, extending our leading strand. Okay? On the opposite strand, we're going to have to be working with Okazaki fragments. And so we're going to start here because we can sort of do it in sequence. We've got, again, helicase opening up, single-stranded binding protein stabilizing, primer being laid down with the help of primase, all right? Once the primer is in place, DNA polymerase 3 starts to elongate off of that primer. Once that's done, we have DNA polymerase 1 removing the primer and replacing it with DNA. And finally, ligase ties it together, all right? And the numbers on the figure here show when these occurred. Obviously, this occurred first, right? And these are occurring subsequently, but I was giving it to you in the opposite order to sort of see how you went about, right, accomplishing that, all right? So, again, mostly, right? Do we have to connect here from our point of origin to whatever's happening on this strand here? Yes. So you're going to need it there as well. All right? So this is an important figure to look at, guys, because you have to recognize from that point of origin. James, I should have just pointed to you here. So recognize then, this is the leading strand, right? But the other side, on the other half of the replication bubble, you're going to have to use Okazaki fragments there, aren't you? Right? And so you've got, beginning at that point of origin, you've got a leading strand on one side of the origin, and on the other side of the origin on that same strand, that is a lagging strand built in pieces. First this one, then this, right? And then likewise on the opposite side. Here we have Okazaki fragments, here we have a leading strand, okay? You should draw this. You should draw this at home so you get it all in your head, all right? So you see this is going to be continuous piece. I'm reading 3 to 5. Here I'm having to read in the opposite direction, so I'm going to have to use my fragments, all right? Or I'm having to build in the opposite direction, all right? And here are those enzymes then that we've just been talking about, okay? The double helix is destabilized with helicase. Single-stranded binding proteins keep the strands apart. Primase builds a primer. DNA polymerase, specifically DNA polymerase 3, adds the new strand. DNA polymerase 1 removes the primer and replaces it with DNA. All right? Ligase ties all the pieces together. And we have one more bit, and that is that DNA polymerase comes back and checks its work. All right? Because you don't want to make a mistake. You don't want to allow a mispairing, which can happen, to persist. So DNA polymerase comes back and checks its work, and we're going to talk about that. Yeah. I don't remember right off the top of my head whether it's something that's carried out by polymerase 3 or whether it's a different polymerase. And the reason I don't remember is that in prokaryotes, it's a different sort of set of polymerases. In eukaryotes, we talk about polymerase 1 and polymerase 3. And I'm sort of putting in your heads DNA polymerase 1 and doing DNA polymerase 3, sort of to give you those two, right? More often than not, we just talk about DNA polymerase as a complex of enzymes that are working on the DNA, and we don't necessarily specify, right? But I'm not answering your question. It's a good question. All right. Okay. All right. So the third thing we said, DNA polymerase goes in and checks its work. When DNA replication is taking place, the rate of mispairings is about 1 in 10,000. All right. So I said nucleotides pair to the template, to the parent strand, spontaneously, A with T, G with C. But there's a 1 in 10,000 chance of a mispairing. When DNA polymerase comes back and checks its work, it catches most of those errors, so the chance of leaving an error behind becomes 1 in a million to 1 in a billion, all right? So that number of a mutation rate, because that's what this is now, we've just found ourselves right at the point of where mutations happen. This is where mutations happen. They are errors that occurred during DNA replication that went unnoticed, all right? So that mispairing chance is 1 in 10,000. The chance of or correction, once it's occurred by DNA polymerase, reduces the error rate 1 in a million to 1 in a billion, all right? And those are, what's left then is the spontaneous mutation rate. Would we hope that DNA could behave more faithfully and leave us with a perfect record? Sounds nice. Would that be an ideal world? DNA polymerase never makes a mistake? No, because there won't be any variation. Mutations are the ultimate source of genetic variation. And though you don't want those mutations for yourself or in your kids, you want the neighbor's kids to get those, right? Right? You want some other member of your species to have that mutation, because chances are it's probably not going to do them any good, but it may. But you don't anticipate the environment changing right away, so you're happy to stay with the status quo, all right? But a tiny mutation rate is absolutely important. It's essential, right, to infuse a little bit of novelty into the genome all the time as a background, all right, as a buffer for environmental change to keep the species alive, okay? So this is an interesting number. It's nature saying, I don't want to be messing with it all the time, right? But a little bit is absolutely essential for survival of the species, and nature has sort of arrived at this mutation rate. Now, as mistakes occur then, as I said, DNA polymerase comes in and corrects those mismatch repairs, all right, along with a number of other proteins. And we see that when people have mutations in the genes for the repair proteins, they have a high incidence of cancers, including colon cancer. Colon cancer I put in particular because we have actually identified patients with colon cancer that have a mutation in those repair proteins, which means that they're not able to detect the errors that occur during DNA replication. So you are very much depending on the efficiency of these repair systems for detecting mutations, all right? And when they don't, why you see higher incidence of cancers. Likewise, let me be clear, and I'll come back to this because I don't want to spill over here longer than I need to. What I've just talked about, mismatch repair, means specifically pairing errors that occur during DNA replication. Is there damage that occurs to the DNA outside of the replication process that needs to be tended to? Yes, and we call that excision repair. So when you put on your sunscreen, you are protecting yourself from ultraviolet radiation that may damage your DNA, not your replicating DNA, but your DNA that's just sitting there in your cells that later, when it gets replicated, is going to be affected because of damage in the DNA. And you want to maintain your DNA healthy so that when damage occurs to your DNA outside of mismatch repair error or mismatches that occur during replication, you have a system for maintaining the health and quality of your DNA. That's called excision repair. I'll say a word about it next time. All right, that was the Nobel Prize in chemistry this past year. All right, was the 3 scientists that worked on studying excision repair. All right, so there you go. All right, and mismatch repair. All right, we'll come back to this briefly on tomorrow. Okay.